Welcome to this lecture on the regulation of gene expression by steroid hormones. This is lecture number 21 in this course on eukaryotic gene expression, basics and benefits. Now, in the last few classes, maybe last 5 classes, we have been discussing about how signaling molecules which interact with membrane receptors ultimately activate or repress transcription of specific genes. That is, how signaling molecules interacting cell surface receptor change or alter gene expression programs inside nucleus. And we have been discussing at least 3 different kinds of receptors. One of them called GPCRs or G protein coupled receptors, which contain the 7 transmembrane domains, and what kind of signaling molecules interact with these receptors and what kind of signaling cascade ultimately leads to activation of gene expression. We also talked about receptor tyrosine kinases, how growth factors bind to growth factor receptors, activate the receptor tyrosine kinases, and how they through the MAP kinase activity lead to ultimately activation or repression of target genes. And in the last class we discussed about signal transducers and activators of transcription, that is how STATs are involved in the regulation of gene expression by cytokines, and there are a number of other cell surface signaling molecules and cell surface receptor pathways, but I gave you these 3 different pathways as examples of how molecules interacting with cell surface receptors can ultimately transduce signals through into the cell, and activation of specific kinases can ultimately lead to the activation of specific transcription factors, and this results in the activation or repression of gene expression. And we also mentioned that there is a lot of crosstalk that goes on between these different signaling pathways, and I gave you some of the examples. What we will do today, we will now go into the inside the cell and discuss about those molecules which can diffuse through the cell membrane and enter inside the cytoplasm and then bind to specific intracellular receptors, and how that results in the activation or repression of specific target genes. One group of molecules which do this are steroid hormones. So, what we will discuss in this class is how steroid hormones, what are steroid hormones, and how steroid hormones enter the cell and bind to specific specific intracellular receptors and activate or repress transcription of specific target genes. So, this is what is the I have shown a schematically here. So far we have been discussing about molecules which bind to specific receptors on the cell surface, but today we are now going to talk about molecules which can diffuse through the cell membrane, bind to specific receptors, and often when these molecules bind to these receptors, it results in the conformational change, and these modified receptors now often dimerize, and these dimerize then go inside the nucleus, bind to specific promoter sequences and regulate gene expression. So, we are going to use steroid hormones as example of this group of molecules and ask the question how steroid hormones are able to do this. That will be the crux of today's lecture. What are steroid hormones? Steroid hormones are steroids that act as hormones. So, steroid hormones can be grouped into 5 groups based on the receptors to which they bind, and these are known as glucocorticoids, mineralocorticoids, androgens, estrogens, and progestogens. And I have just given you the structure of here. Cortisol is a glucocorticoid which is mainly made in the adrenal gland. Aldosterone is a mineralocorticoid again made in the adrenal gland. Testosterone is a male sex hormone which is made in the testes. Estradiol and progesterone are the female sex hormones which are made in the ovaries. So, let us now, and you can see the moment I tell you the names, these are all very important molecules. They control a number of very important cellular processes, and they all have more or less similar structure. As you can see, they are derived from cholesterol. So, let us now try to understand what are these molecules and how these molecules bring about their physiological effects. This is the structure of cortisol I have shown here. They are derived from cholesterol, and this is a glucocorticoid which is synthesized from progesterone in the zona fasciculata of adrenal cortex. The adrenal gland has 2 major regions called the cortex and medulla, and the glucocorticoids are made from the adrenal cortex, whereas the epinephrine and norepinephrine, which we discussed in the previous class, which are the molecules which interact with cell surface receptors, they are made from the adrenal medulla. Now, cortisol is involved in stress adaptation, elevation of blood pressure, sodium uptake, and numerous effects of immune system, including anti-inflammation. Many of the drugs which are available in the market to reduce inflammation are glucocorticoids. So, glucocorticoids act as major anti-inflammatory agents. Aldosterone, again as I said, it is a mineralocorticoid. It is produced from progesterone again in the zona glomerulosa region of the adrenal cortex. Again, it is involved in the raising of blood pressure, adjusting regulation of the fluid volume in the blood, and also in the sodium ion uptake. So, plays a very important role. Progesterone is a female sex hormone. It is produced directly from the pregnenolone and secreted from the corpus luteum of the ovary. It is responsible for changes associated with the luteal phase of the menstrual cycle and is involved in the differentiation of mammary glands. A very important hormone in the case of females. Testosterone is a male reproductive hormone. It is an androgen, and it is synthesized in the testes and responsible for all the secondary male sexual characteristics. Estradiol is again a female hormone, again produced from the ovary, and is responsible for the female sexual characteristics. So, as you can see, these 5 hormones are very important. They have very important biological functions, and they are all synthesized from cholesterol. I have just shown here a slide that is taken from this particular website, which tells you how the glucocorticoids and mineralocorticoids, namely the cortisol, corticosterone, which together constitute the glucocorticoids, aldosterone and androstenedione, which together constitute the mineralocorticoids, and how they are synthesized from cholesterol, and what are the various enzymes involved in this pathway. Ultimately, these molecules are synthesized. We will not go into details of how they are made, but just know that the glucocorticoids and mineralocorticoids, examples cortisol and aldosterone, they are synthesized by the adrenal gland, and they are all derived from cholesterol. Similarly, the gonadal hormones, both estradiol and progesterone, as well as testosterone, again cholesterol is the precursor for the biosynthesis, and here is the pathway by which cholesterol is converted into the testosterone or estradiol. Need not remember the pathway at this stage, but just to tell you that they are structurally very similar molecules, but they have very different physiological functions, and they are produced in different regions of the body. These are natural steroid hormones that we have talked so far about. There are also what are called as synthetic steroid hormones, again play a very important role, has a lot of biomedical applications. They are variants of the structure that we have just seen here, and they have either more potent agonist or antagonistic action. Mostly agonistic action. The antagonists also have very important effects. Glucocorticoids, synthetic glucocorticoids, for example, is called prednisone, dexamethasone, triamcinolone, and so on and so forth. Synthetic mineralocorticoids, fludrocortisone, these are all derivatives of the basic structure which I have shown in the previous slides. Many androgens, in fact, they have very important role, especially those athletics. For example, athletes who want to have more muscles, who want to have a who want to win races much faster, they take what are called as anabolic steroids, which are actually banned. Athletes are not supposed to take these anabolic steroids. What is called as a dope test, and if you test positive in this dope test, you cannot participate in many of these competitions. And these anabolic steroids are nothing but synthetic steroid hormones, especially synthetic androgens, oxandrolone, nandrolone, and so on and so forth. Very important in the sports area. Estrogens, again you have a synthetic estrogen, diethylstilbestrol called DES, and you have synthetic progesterones, norethindrone, medroxyprogesterone acetate, and so on and so forth. So, these steroid hormones have very important biomedical applications because they are very potent molecules that alter the physiology. Now, steroid hormones have been around for a long time. In fact, steroid hormones have been known to exist since the early 20th century. However, it is only in the early 1960s and especially in the late 1980s the molecular mechanism of action of the steroid hormones really became very clear. So, it is not until early 1960s that the idea of specific hormone binding molecules in the target tissue of these hormones began to emerge. So, till 1960 people did not know how these hormones are working, but then when biochemistry, early 1960s and 70s, the biochemistry ruled the world in the biology, and when biochemist starts grinding the tissues and started looking what kind of protein molecules bind to the steroid hormones, it soon became very clear tissues which are responsive to these hormone molecules actually contain proteins which bind to these hormones, steroid hormones. So, the concept that these hormones may be acting by actually binding to specific receptors began to emerge by biochemical studies that carried out in the early 60s. The analysis of the steroid hormone receptors had relied purely on biochemical techniques. As I said, one of the major methods by which people used to study steroid hormones in the late 1960s and early 1970s is you use a radio labeled steroid hormone, and then you see how the radio labeled hormone goes and binds to proteins inside the cells, and then try to monitor, do the kinetics of binding, and then see where this receptor is localized, and so on and so forth. So, primarily biochemistry contributed a lot to understanding the molecules or the protein molecules which actually bind to the steroid molecules in the early 60s. But it is only after the genes encoding these receptors were cloned, it became possible to carry out detailed studies of various functional domains of the receptor. So, the best that biochemists could do is to see what kind of molecules bind and what is the affinity of these steroid hormones to these molecules, and with great difficulty they could actually purify some of these receptor molecules, and in the late 70s some of these proteins or the receptor molecules could also be sequenced. So, the partial amino acid sequence of some of these steroid hormone receptors were available, but complete characterization was not possible using just biochemistry alone. But the late 70s and the early 80s was the dawn of molecular biology. It became to clone genes, it became to identify the amino acid sequence of proteins using the gene sequence and so on and so forth, and once restriction enzymes, cloning techniques became available, that made a very major role in understanding the function, structure and function of steroid hormone receptors. So, what I am going to go through is to take you through a brief historical perspective of what kind of people have actually contributed a lot to the understanding of the steroid hormone receptor structure and function, and what is the historical perspective of this field. That is what we will discuss in the next few slides. Now, as I said, biochemistry played a major role in understanding the structure and function of steroid hormone receptors. For example, in the 1968 some of the studies actually led where you take a tissue, homogenize the tissue and then do a differential centrifugation by which you can separate nuclei, you can separate cytoplasm, and then they took these different fractions and see where is the protein which is able to bind to the steroid hormone is present, and based on such studies people actually showed that when there is no hormone, this binding protein is actually present in the cytoplasmic fraction, but the moment you add hormone, this activity shifted to the nucleus. So, these kinds of biochemical studies started giving an idea of how these molecules actually act or how these molecules are able to bring about physiological response, and they clearly told that these molecules actually enter inside the cell and bind into specific intracellular proteins. Again, once people started purifying using these kinds of technical localization studies and again using biochemical purification techniques, people have started purifying these receptor proteins from some of these tissues, and once you got a purified receptor, you could actually do a partial tryptic digestion of this receptor or find out the internal amino acid sequencing or the internal peptide sequencing, and based on these peptides as well as the purified receptor, you can immunize either mice or rabbits and you can generate antibodies. So, once you have antibodies which are specific for these kinds of steroid hormone receptors, then people use these antibodies to ask the question where exactly this receptor is present. For example, if you have a specific tissue which has a binding protein for that particular steroid hormone, you do what is called as an immunofluorescence technique, where you add a receptor antibody which is specific to that particular receptor, and then you have what is called as a second antibody which is always conjugated to a fluorescent molecule. It could be what is called as a fluorescent isothiocyanate or TRITC, and now we have so many other modern dyes which under a fluorescent microscope either give a blue color, a green color or a red color. So, using these kinds of immunofluorescence techniques, people and using receptor specific antibodies, people could actually demonstrate that these receptors, when there is hormone inside the cell, they are actually present inside the nucleus. So, a picture began to emerge that these steroid hormones basically act by binding to certain receptor molecules present inside the cytoplasm, and once the hormone binds the receptor, this receptor is going to the nucleus and probably it is activating the transcription of specific genes, and that is how these steroid hormones are able to bring out specific biological effects. This is the paradigm that began to emerge many of these biochemical studies that was carried out in the late 1960s. So, this is what I have summarized what I just told you here. It is generally thought that unoccupied steroid receptors can exist in the cytoplasm, while the occupied receptor act in the nucleus on specific target DNA sequences. But this is all again not much experimental evidence is available, it is mostly conjectural. So, when bound to the hormones, your cytoplasmic steroid hormone receptors move to the nucleus, and the purified receptors probably are going and binding to specific DNA sequences. Some of the key experiments which were done again using biochemistry and a little bit of molecular biology, because as I said, the late 70s and 1980s is the one that was the dawn of molecular biology, and a number of molecular biology techniques became available, and people started using, especially biochemists who were actually doing protein purification and protein characterization techniques, now realized that they could answer many of these questions using many of these molecular biology techniques by looking at the transcription, by looking at the genes and so on and so forth. So what, for example, Keith Yamamoto's group actually did in 1983, they published a paper in Cell in 1983, sequence specific binding of glucocorticoid receptors in the MTV DNA at sites within and upstream of the transcribed region. So there is a virus called mouse mammary tumor virus, and this virus responds very well. For the transcription of this virus, you require glucocorticoids. So people like Yamamoto started asking the question, how does this mouse mammary tumor virus promoter is getting activated by glucocorticoids? Because glucocorticoid is producing in our body, and here is a mammalian virus, and the transcription of this virus is actually getting activated by a hormone which is produced by our body. So what they did, they actually took the what is called as the long terminal region, long terminal repeat or LTR of this particular MMTV virus or MTV virus, and then asked the question, where exactly does the glucocorticoid receptor bind? Because by this time it is very clear, all the effects of glucocorticoids are probably modulated through the glucocorticoid receptor. So if the glucocorticoids is activating the expression of this mouse mammary tumor virus, people ask the question, the glucocorticoid receptor must be doing something to the virus, and the first question they ask is, does it go and bind to specific DNA sequences within the mouse mammary virus tumor DNA? And in fact, he actually showed that there are about 5 regions within this MTV DNA that are specifically bound by purified glucocorticoid receptor, and one resides upstream of the transcription start site, and others are distributed within the transcript sequences between 4 to 8 kb from the initiation site. So for the first time, studies like this have actually shown that here is a receptor, mammalian receptor, that is actually binding to some specific sequences within this viral genome, and this binding probably is responsible for activation of expression of the viral genes. So such studies with purified steroid hormone receptors demonstrated that they are likely to be sequence specific DNA binding proteins, and they are likely to be transcription factors. So this is what emerged from many of these biochemical studies. So classical ligand receptor interaction studies, understanding their kinetics of binding and affinity of these ligands to these receptors, as well as biochemical purification techniques, followed by cellular localization using immunofluorescence, as well as and more on the late 70s and early 80s using some of the molecular biology techniques. It became very clear that one of the major mechanisms by which the steroid hormones are acting is by binding to the steroid hormone receptors, and these steroid hormone receptors are then going inside the nucleus and binding to specific regions within the DNA sequences, and that is how they are able to bring out specific physiological effects. So this is the paradigm that emerged before the molecular biology era started. The most important phase in the study and understanding of the steroid hormone receptor function began with the cloning of the steroid hormone receptors. So I am going to give you a brief historical perspective of how the steroid hormone receptors were cloned. Now studies from a number of laboratories, such as those headed by Elwood Jensen, Bert O'Malley, Keith Yamamoto, and many others. I am not going to quote everybody who worked in those areas. These are some of the very important people who made very important contributions to the understanding of steroid hormone receptor functions. All these studies actually led to the hypothesis that activated steroid hormone receptors go and bind to specific DNA sequences in the nuclei of the target cells, and this is what induces the transcription of these genes, leading to specific production of specific proteins. So all the biochemical studies and immunolocalization studies and typical receptor ligand interaction studies, all these things pointed out that when you add a hormone to this steroid hormone to the cell, it is entering the cell, binding to this intracellular receptor, and then it goes inside the nucleus, and this binding of the receptor to specific DNA sequences results in the synthesis of specific proteins, and that is how the steroid hormones are bringing about their physiological responses. Now by the end of 1980s, when techniques for cDNA library scanning and cloning and sequencing of DNAs became a routine, the first receptor cDNA encoding glucocorticoid receptor was cloned by Ronald M. Evans in the Salk Institute in La Jolla, California, United States of America. So 1980s, as I told, was the golden era of biology, because the molecular biology techniques became available. People realized that you can actually purify messenger RNAs for a specific protein, encoding a specific protein, and you can derive a cDNA of those, and you can actually clone the cDNAs. So from millions and millions, hundreds and thousands of genes which are available, you can actually pull out a specific gene or specific cDNA coding for a particular protein. So once the creation of cDNA libraries and how to screen the cDNA libraries and how to isolate a specific cDNA became available, people began to understand, can we clone a cDNA that codes for, or can you clone the messenger RNA that actually synthesizes these steroid hormone receptors. And the first one was done by Ron Evans in Salk Institute in the early 1980s. Once the receptor for glucocorticoid receptor was cloned, this was soon followed by cloning of the cDNAs coding for estrogen, progesterone, androgen, so on and so forth in the subsequent years. So, I am going to spend some time to tell you how actually all these things were done, because these are landmark papers in the area of steroid hormone receptor structure and function. So, let us now see what exactly they did. So, the first paper on the characterization of the cDNA encoding the glucocorticoid receptor was published in the journal Nature in 1985-1986 by Ronald Evans group in Salk Institute, and is entitled Primary Structure and Expression of a Functional Glucocorticoid Receptor cDNA. This I would consider as a landmark paper in the area of steroid hormone receptor structure and function, because this marked the beginning of a new era in the biochemistry, and this paved way, or this probably is primarily responsible for our current understanding of the steroid hormone receptor structure and function. So, what this group actually did is to demonstrate that there are two forms of glucocorticoid receptor cDNAs. So, what basically they did, they took the RNA isolating from a tissue which expresses glucocorticoid receptor, converted them into cDNA by using reverse transcriptase and so on, and then took the cDNAs of all these messenger RNA that are synthesized in the tissue and then put them in a phage vector and then made a library. So, I basically made a cDNA library, and then using either antibodies specific for glucocorticoid receptor or using oligonucleotides which specifically, or degenerate oligonucleotide that corresponds to the specific amino acid sequences of this glucocorticoid receptor which was available at the time, because the purified receptor could be cleaved and the partial amino acid sequence could be identified for some of the peptides, using either oligonucleotides that correspond to those peptide sequences or by using antibodies raised against the purified glucocorticoid receptor. You basically screen these libraries and ask the question, which phage clone harbors the cDNA that actually codes for the glucocorticoid receptor. So, by cDNA screening, cDNA library screening, they have actually identified two clones, and one of them coded for a glucocorticoid receptor of 777 amino acid, and they call it as glucocorticoid receptor alpha. Another coded for a 742 amino acid protein which they call as a glucocorticoid receptor beta. And you can see this is the current knowledge of our glucocorticoid receptor structure, and the foundation for this was actually laid in the year 1995-86 by Ronald Evans group, and we now know that all these amino acids are encoded from about 9 exons. These are marked from 1 to 9 here, and today we know that the glucocorticoid receptor structure actually consists of what is called as a N-terminal domain, and it consists of a DNA binding domain which actually comes from exons 3 and 4, and it consists of what is called as a ligand binding domain to which the glucocorticoid goes and binds. So, the alpha is up to this, whereas the beta is truncated here. Now, the same year, from the same group, it also became another important landmark paper was published, where it was reported that the domain structure of the glucocorticoid receptor and its relationship to VRBG and its relation to VRBG oncogene product. Now, you can see here the importance of molecular biology. Now, the biochemical purification studies could only at the most get partial amino acid sequence, or they could raise antibodies, but nobody could obtain the full length amino acid sequence of glucocorticoid receptor using conventional biochemical techniques, because it is impossible for you to sequence the entire how many amino acids, almost 777 amino acids. You cannot do typical amino acid sequence of this protein. It is too Herculean a job. But once the cDNA sequences are available, from the cDNA sequence you can actually deduce the amino acid sequence, and based on the amino acid sequence people ask the question, what kind of a protein is this? What kind of functional domains are actually present in this glucocorticoid receptor? And based on such studies, in the subsequent paper they actually shown that this glucocorticoid receptor, some of the domains actually resemble to that of an oncoprotein called VRBA. Now, what is this VRBA? The VRBA is an oncogene product of a virus called avian erythroblastosis virus, or AEV. So, these are viruses which, when we infect, it actually causes cancer, and one of the major proteins which is responsible for causing cancer in this virus is a protein called VRBA. And what the studies of this group actually show is that the structure of the glucocorticoid receptor very much resembles the structure of this oncoprotein. So, you can see people who have been studying steroid hormone suddenly realized there is some relationship between steroid hormone receptors and cancer, because here is a viral protein which is causing cancer, and the structure of this protein resembles very much that of the glucocorticoid receptor. So, people ask the question, what is the link? So, they predicted in this paper that the oncogenicity of this virus may result in part from the inappropriate activity of a truncated steroid receptor or a regulatory molecule encoded by VRBA. So, the moment they found that structurally these two proteins are related, people realized that the mechanism by which this oncoprotein is acting causing cancer may be similar, may be because of inappropriate signaling. So, they suggested that a mechanism may be, they suggested this is a mechanism by which transcription factors may facilitate transformation. So, these kinds of studies clearly show that transcription factors may be involved in cellular transformation leading to cancer. And they also identified another short region in the human glucocorticoid receptor which had very high degree of homology to certain proteins which are involved in the regulation of development, like the Drosophila homeotic proteins encoded by Antennapedia, Fushitarazu, etc. Now, we will come back to this transcription factor regulation in development in subsequent classes, but what I am trying to tell you in this paper is that once the cDNA for the glucocorticoid receptor was cloned and the domain structure was analyzed, it became very clear that this is likely to be a transcription factor. There is something look like a DNA binding domain, and it also had a homology to a very important viral protein which causes cancer, and people realized that the mechanism by which this virus are causing cancer may be because of inappropriate activation of some of the genes which are actually activated by steroid hormone receptors. Now, they soon realized that in the subsequent year, two groups actually published a paper where they actually demonstrate the VRBA gene is nothing but a thyroid hormone receptor. So, the link between cancer and hormones became firmly established when they realized that one of the viral oncoproteins, which very much resembles that of a glucocorticoid receptor, actually codes for a thyroid hormone receptors. And I can see I have just highlighted some of the important findings from this paper, where this group again from Ronald Evans' lab actually showed that the CRBA gene encodes a thyroid hormone receptor. And what they showed is that the cDNA sequence of human CRBA, the cellular counterpart of viral VRBA, indicates that the protein encoded by the gene is related to steroid hormone receptors, and binding studies with the protein show it to be a receptor for thyroid hormone. So, they took the gene coding for the thyroid hormone receptor, did what is called as in vitro transcription and translation, and that is you take the RNA, translate it in vitro, and this in vitro translated protein was able to bind to thyroid hormone. So, here is a viral oncoprotein involved in cancer that seems to be binding to thyroid hormone, clearly saying that there is a link between hormone signaling and cancer. Again, around the same you can see these two papers are published in the same issue of Nature back to back. This is from Wenstorm's lab in Europe, where again he showed that hormone binding localization of CRBA protein suggests that it is a receptor for thyroid hormone, a nuclear protein that binds to DNA and activates transcription. So, the product of viral oncogene is defective in binding to the hormone, but still localized to the nucleus. So, you can see here is a viral oncoprotein which binds to the DNA, but it does not bind to thyroid hormone, clearly indicating that if you have receptors which are defective in ligand binding or which are hormone binding, and such receptor may cause inappropriate signal transduction pathways, activate inappropriate target genes, and this is what can lead to ultimately cancer. So, these kinds of studies established a link between virus induced cancers or viral oncogene products and hormone signaling pathways. So, the cloning of glucocorticoid receptor around the same time when Ron Evans group cloned the glucocorticoid receptor, a number of other laboratories started cloning other receptors, because they all had either antibodies of specific receptors or they had the partial peptide sequence, amino acid sequence of certain peptides of these various receptors. And Pierre Chameau's group in France, for example, took the antibodies against the estrogen receptor, as well as they made oligonucleotides against peptide amino acids of specific peptides of the estrogen receptor, and they cloned the estrogen receptor cDNA. So, they reported the cloning of the cDNA for estrogen receptor in the breast cancer cell line FNF7, and the expression of cDNA in HeLa cells produce a protein that has the same relative molecular mass and binds estradiol with the same affinity as MCF receptor. So, it became very clear the protein which is actually produced from this cloned gene is more or less the same as the estrogen receptor that is expressed in a well known breast cancer cell line. So, there is they also reported that there is extensive homology between the estrogen receptor and the RBA protein of the oncogenic avian erythroblastosis virus. So, all this became clear that whether you clone a glucocorticoid receptor, whether you clone an estrogen receptor, they all seem to have similar structure, and the structure is very similar to that of an oncoprotein encoded by a oncovirus or a tumor virus. So, all these studies clearly led to the proposal or a hypothesis that there exists a super family of oncogenic hormone receptors. So, there are certain oncoproteins which are highly homologous to the hormone receptors, and these superfamily of receptors may be actually involved in viral transformation. So, both Chambon as well as Ron Evans group actually proposed that these human steroid receptors as well as some of these viral oncoproteins like RBA may actually constitute a superfamily of enhancer binding proteins, and they may actually cause cancer. Once the glucocorticoid receptor cDNA and the estrogen receptor cDNA was cloned, subsequently a number of other steroid hormone receptors were also cloned. For example, between 1960s and 1968, for example, the chicken progesterone receptor cDNA was cloned as well as the human androgen receptor cDNA was cloned. So, once all the cDNA for the steroid hormone receptors were cloned, it became very clear that they all encode a superfamily of hormone receptors. This I am just putting a slide which I have already explained here. What was the strategy that was employed for cloning all these genes encoding steroid hormone receptors? The strategy is to make a cDNA library, that is, you isolate messenger RNA from all these tissues which are expressing the specific receptors, and then you convert all this messenger RNA into cDNA, and then you clone these cDNAs into a phage vector and make a phage library or a phage cDNA library, and then you take this phage cDNA library and either you probe them with antibodies against specific receptors which have been purified from various cell types, and you take these antibodies raised against purified receptors, or you make oligonucleotide degenerate oligonucleotides corresponding to the amino acid sequence of specific receptors, and using either radio-labeled oligonucleotides or using antibodies you can screen these phage libraries, phage cDNA libraries. That is how they could isolate these cDNA clones, and then you sequence the cDNA clone and deduce an amino acid sequence from the cDNA and see what kind of a protein that they are coding for. So, this was the strategy that was used for isolating the various steroid hormone receptors in the early 80s and late 1980s. So, once all these studies were done, so you had the receptor for glucocorticoid receptor, you had the receptor for estrogen receptor, thyroid hormone receptor, and so on and so forth. It became clear from all these studies that all these receptors have certain common structural domains. For example, all these receptors have what is called as a ligand binding domain, which probably binds the hormone. It is called as a hormone binding domain or the ligand binding domain. The receptors also had what is called a DNA binding domain in the middle, and this probably is responsible for binding to specific sequences, and it also had what is called an amino terminal hypervariable region, which was not that well conserved between various receptors that they had cloned so far. So, some of the three major functional domains of these receptors were a ligand binding domain, a DNA binding domain, and a hypervariable amino terminal domain. So, these were the major domains which are present in all these receptors. But today we now know that there are much more detailed information is available on the various domains of the steroid hormone receptors. For example, we now know that this amino terminal region, which is actually shown as A/B, actually contains what is called as an activation function 1. It actually is responsible for ligand independent activation of steroid hormone receptors. And we also know that the region known as C actually codes for a DNA binding domain, and this DNA binding domain consists of 2 zinc fingers. We had discussed about zinc fingers in the previous classes when we talked about the structure and function of various DNA binding proteins, and the steroid hormone receptor contains 2 C2H2 type of zinc fingers, and we will look at the structure in a few more minutes. And it also became very clear, people started looking at the mechanism by which this DNA binding domain acts, and I will discuss in more detail in a few more minutes. And this DNA binding domain contains what is called a P box and what is called as a dimerization region, and plays a very important role in the DNA binding function of the steroid hormone receptor. And as I said, all these steroid hormone receptors are present in the cytoplasm when there is no hormone, and they go into the nucleus in the presence of hormone. So, they contain what is called a nuclear localization signal, and this nuclear localization signal is masked when there is no hormone, and once the hormone binds, it induces a conformational change. So, the nuclear localization signal gets exposed, and therefore the receptor goes inside nucleus. It also has a ligand binding domain. The ligand binding domain has 2 important functions. It has a ligand dependent activation function, that means it has an activation function 2, whereas the one in the amino terminal domain is referred as activation function 1, and when ligand binds, this activation function is activated, and now this in the presence of ligand, the receptor is able to interact with the general transcription machinery and the RNA polymerase, resulting in the activation of transcription. And the domain also, in the absence of ligand in certain receptor, can act as a repressor. So, the cloning of these cDNAs of these various receptors paved way for a very detailed understanding of the various functional domains of these various steroid hormone receptors. Now, once all these receptors were cloned, there are also many, it also became clear that the structure of the steroid hormone receptors bared homology to other non-steroidal receptors. For example, thyroid hormone receptor, thyroid hormone is not a steroid hormone, but the receptor for thyroid hormone was very homologous to that of a glucocorticoid receptor or an estrogen receptor. So, people soon realized that there exists a nuclear receptor superfamily, and this nuclear receptor superfamily contains not only steroid hormone receptors, but also contains other non-steroid hormone receptors or other hormone receptors and which are not steroids. So, all these based on the structural homology were grouped under one nuclear receptor superfamily. So, by sequencing cDNAs of various hormone receptors and deducing amino acid sequences, all these were grouped under a single nuclear receptor superfamily, and two broad categories of these receptors exist in this. One are called as the type 1 receptors and is called the type 2 receptors. The type 1 receptors basically consist of the steroid hormone receptors. They undergo nuclear translocation after ligand activation and bind as homodimers to inverted repeat DNA sequence hop sites referred to as the hormone response elements. So, the DNA sequences to which the steroid hormone receptor binds is known as HRE or hormone response element. Glucocorticoid will bind to glucocorticoid response element, estrogen will bind to estrogen response element, thyroid hormone will bind to thyroid hormone response elements, and so on and so forth. So, in general they are known as hormone response elements. So, the example for the type 1 receptors are receptors activated by steroid ligands such as glucocorticoid receptor, mineralocorticoid receptor, estrogen receptor, progesterone receptor, and androgen receptor. So, all these steroid hormone receptors are classified as type 1 receptors because usually they stay in the cytoplasm in the absence of the hormone, and once the hormone binds they undergo conformational change, then go into the nucleus and often bind what is called say inverted repeat sequences. We will come to that in a minute. Whereas the type 2 receptors, they often are retained in the target cell nucleus regardless of the presence of ligand. So, unlike the classical steroid hormone receptors which are present in the cytoplasm when there is no hormone and goes into the nucleus only when you have hormone, the type 2 receptors are already present in the nucleus irrespective of whether the hormone is there in the cell or not. So, they are nuclear receptors, whereas steroid hormone receptors are cytosolic in nature in the absence of hormone and nuclear in nature in the presence of hormone, whereas the type 2 receptor are always nuclear in the nature. The function of ligand is actually to unfurl or the activate the transcription activation function of the type 2 receptor. So, in the absence of hormone, although the receptor is bound to the DNA, it does not activate transcription, but once the hormone binds, it induces a conformational change, therefore, the receptor can activate transcription. Example for type 2 receptors are receptor for thyroid hormone, retinoic acid, vitamin D, and so on and so forth. We will discuss type 2 receptors in the next class, but today we will confine ourselves to only what is called say type 1 receptors, which basically comprise of the steroid hormone receptors. Now, so it became very clear that these steroid hormone receptors act or bring about their physiological effects by primarily binding to what are called the hormone response elements which are present on the promoter regions of various genes. So, once the hormone binds, the intracellular receptors now function as transcription factor. They go and bind to the hormone response elements in the promoter regions of various genes, and this is how they act transcription of various genes. So, all the genes which are the targets for glucocorticoid hormones, they invariably contain a hormone response element in the promoter region, and that is how the transcription of these genes are getting activated in presence of those hormones. So, if the target gene is to be activated by glucocorticoids, then it should contain the glucocorticoid response element. If the target gene is to be activated by an estrogen, it should contain estrogen response element, and so on and so forth. So, by having specific hormone response elements in the promoters of these target genes, these hormones through their receptors are able to bind to these sequences and bring about transcriptional activation. So, this became the general mechanism by which steroid hormones bring about transcriptional activation. Now, one of the important experimental techniques that actually paved way for understanding the structure and function of these nuclear receptors or the steroid hormone receptors is an assay called as cis-trans co-transfection assay. It is because of this assay it became possible to identify the various functional domains of these steroid hormone receptors. I have already discussed the cis-trans co-transfection assay in one of the earlier classes, especially in the introductory classes of this course, but I do not mind repeating again. Basically what this actually means is that This is a transient transfection based hormone response element reporter assay developed again in the laboratory of Ronald Evans at Salk Institute, and it became the tool of choice for investigating the mechanism by which nuclear receptors regulate the target of genes. So, it is a very important assay, and because of this assay that people could identify the various functional domains of steroid hormone receptors. Now, what is this? In this assay, a cDNA encoding the receptor or part of a receptor is transfected into a suitable cell line along with a reporter gene. Usually it can be luciferase gene linked to a promoter controlled by one or more of the hormone response element specific for the receptor being studied. So, basically what you do in this assay is that you take the cDNA that codes for, let me say I want to see whether glucocorticoid receptor is actually activating from a promoter or not, or whether a promoter actually contains a glucocorticoid response element or not. So, what I do, I take the promoter sequence, or I take the sequence which is suspected to be a glucocorticoid response element and link it to a luciferase gene which is a reporter gene. It can also be beta galactosidase or it can be chlorophenol transferase. There are a number of such reporter genes. So, you take the luciferase gene, put it downstream of this suspected promoter which is responsive to glucocorticoid, and put this plasmid inside the cell. This is called the cis vector. Now, we take another plasmid which contains, let us say for example, a well known viral LTR, viral promoter, which is known to be expressed by host transcription factors in a number of cell lines. So, you clone the receptor for your glucocorticoid receptor under this promoter, so that when you put this plasmid inside the cell, the transcription factors present inside the cell will go and bind to the promoter and they express the receptor. And once the receptor is expressed, the receptor will now go and bind to the cis vector, the promoter region of cis vector, and now when you add ligand, it will activate transcription. So, if the cis plasmid contains the sequence for binding of the hormone receptor, and then when the hormone receptor is expressed from the trans plasmid, it will go and bind to the luciferase vector and induce the expression of the luciferase gene, and you can then measure the luciferase activity, and then you can now demonstrate that yes, this receptor can actually activate transcription from a specific region. So, by using this assay, not only you can identify a hormone response elements in the promoter region of any gene, you can also dissect out what region of the receptor is important. For example, you can make mutations in the DNA binding domain and ask the question, can it now go and bind to DNA? Can it now activate transcription? Or you can change the amino acid sequences within the DNA binding question and ask the question, how is the specificity of this receptor is domain? Or you can switch the ligand binding domains. For example, since I have the cDNAs for estrogen receptor, I have the cDNA for glucocorticoid receptor, I can take the ligand binding domain of the glucocorticoid receptor and fuse it to estrogen receptor. Now, if I take this chimeric receptor and put it inside the cell, and since the glucocorticoid receptor now has an estrogen receptor cDNA, now it will activate transcription from glucocorticoid response element in response to estrogen, but not in response to glucocorticoid. These kinds of experiments done using this assay clearly indicated that these steroid hormone receptors have a modular structure. You can take the DNA binding domain of glucocorticoid receptor and put it on estrogen receptor, or you can take the DNA binding domain of estrogen receptor, put it inside a glucocorticoid receptor, and when you do this, they start activating from each other response elements. That is, the glucocorticoid receptor containing estrogen receptor DNA binding domain will now start activating transcription from an estrogen response element. Or you can do the other way also. You can take the ligand glucocorticoid receptor binding domain or the ligand binding domain of glucocorticoid receptor and switch it with that of the estrogen receptor, and now they start activating transcription with each other ligands. So, these kind of cis trans-co-transfection assays paved way for characterization of the DNA binding domains as well as ligand binding domains of the various steroid hormone receptors. So, such studies actually demonstrated that steroid hormone receptors actually bind to hormone response elements, and this actually led to the characterization of the various steroid hormone response elements. For example, hormones such as progesterone, androgen, glucocorticoids and mineralocorticoids, they all seem to bind to a specific to the same kind of response element containing AGA, ACA separate and TGT, TCT separated by 3 nucleotides. This is actually called as a inverted repeat. You can see AGA, ACA on the opposite strand it is as TGT, TCT. So, I have AGA, ACA and the opposite strand also has AGA, ACA, and therefore is called an inverted repeat. So, AGA, ACA, TGT, TCT on the opposite strand will again read as AGA, ACA, TCT, TCT. So, you have the same AGA, ACA sequence on the 2 sides of the double stranded DNA. So, these are called as the palindromes or inverted repeat sequences. So, many of the steroid hormone receptors bind to the inverted repeat sequences. So, it became one of the hallmarks of hormone response elements. And interestingly, all these 4 receptors, progesterone receptor, androgen receptor, glucocorticoid receptor and mineralocorticoid receptor, they all bind to a similar sequence which consists of what are called as 2 half sites. the AGA ACA is one half site, TGTT is another half site, and these two half sites are separated by 3 nucleotides. It can be any nucleotide. So when you have this kind of a sequence in the promoter region of a gene, you can say this is likely to be activated by any one of these hormones. Similarly, if you take estrogen receptor actually binds to a sequence called AGG-TCA 3 nucleotides TGA-CCG. So you can say the only difference between estrogen receptor or estrogen response element and a glucocorticoid response element is 2 nucleotides. So if the sequence is AGA ACA, it becomes a glucocorticoid response element. Instead of AGA ACA, if it is AGG TCA, it becomes an estrogen response element. So you can see the physiological effects of estrogen is quite different from the physiological effects of glucocorticoids. But if you see at the molecular level, these two, there is very fine difference that distinguishes between the estrogen receptor and progesterone receptor target genes. So if the target genes in the promoter contains AGA, ACA, TGTCT, it becomes responsive to glucocorticoid. If the same sequence instead of the 2 A's, if it is replaced by a GT, it now becomes responsive to estrogen receptor. So this is a very fine difference exists at the molecular level between genes that are responsive to glucocorticoids and genes that are responsive to estrogen. And very fantastic experiments are actually done to further characterize these DNA binding properties of the steroid hormone receptors. So it became clear that all these steroid hormone receptors, whether it is GR, MR, PR, ER or AR, they all contain 2 zinc finger domains, and this zinc is coordinated by the 4 cysteines. So they contain the C4 type of zinc fingers and it has been very well shown that the DNA binding domain actually contains 2 very important regions called as P box and D box. The D box refers to a dimerization box or a distal box, and P box is the proximal box, and a number of people actually could do mutations within these DNA binding domains and ask the question, what are the amino acid residues which are important for the DNA binding. For example, if you now mutate this particular A adenine in this DNA binding the alanine in this region, if you mutate that to threonine, now this receptor can no longer dimerize. So if you mutate the alanine residue to that of threonine in this amino acid position 458 in the glucocorticoid receptor, now the glucocorticoid receptor cannot dimerize. Now I can just go back and tell you here, since the hormone response elements consist of a palindromes, that means you have 2 half sites, AGA ACA on one strand and AGA ACA on the other strand, and therefore the steroid hormone receptors, in order to activate transcription, they go and bind as dimers. So one monomer recognizes one half site on one strand and the other monomer recognizes on the other strand, and in order for them to bring together, you need a dimerization domain, and there is a dimerization interface in the DNA binding domain, and there is also a dimerization interface in the ligand binding domain of this steroid hormone receptors. So using these kind of cis-trans co-transcription assays, people have identified what are the important amino acid residues that play a very important role in DNA binding. And like I said, in the case of glucocorticoid receptor, one mutation actually can abolish the dimerization function of the receptor. A very important experiment that was done in the 1989, the year 1989, by 2 groups, one headed by Gordon Ringgold's lab as well as Ron Evans' lab in Salk Institute. They actually demonstrated by making specific mutations within this DNA binding domain, you can actually change the specificity of one receptor to the another. For example, in this paper published in Cell, where they actually showed 2 amino acids within the knuckle of the first zinc finger specify DNA response element activation by the glucocorticoid receptor. What they actually showed in this paper is that the ability of the glucocorticoid estrogen receptors to discriminate between the closely related response elements resides in 2 amino acids located between the 2 cysteines in the C-terminal half the first finger. I told you the glucocorticoid response element is AGA ACA half site, whereas the estrogen receptor instead of AGA ACA is AGG TCA. So there are only 2 nucleotides which are different. So what this paper actually showed is that if we change 2 amino acids in the DNA binding domain of the glucocorticoid receptor, instead of recognizing the glucocorticoid receptor, it now recognizes an estrogen receptor. A similar paper was published in the same issue of Cell and this is the amino acid, but again from the Ron Evans group, again the same issue of Cell, where actually they showed is that by simply changing a glycine residue to glutamate in the base of the first zinc finger in the P box, you can now convert a glucocorticoid receptor to that of estrogen receptor with respect to DNA binding specificity is concerned. So if we simply mutate one amino acid, glycine, to glutamate, instead of this receptor recognizing a glucocorticoid response element, it will go and recognize a estrogen response element. So you can see you have a number of steroid hormone receptors, from the gross structure is the same, they all contain a zinc finger domain. but minute amino acid differences between these receptors actually distinguish whether this receptor will go and bind to a glucocorticoid response element or whether it will go and bind to a estrogen response element. So, palindromics and all steroid hormone response elements exist as palindromic sequences, and the receptor actually goes and binds as a dimer, as is shown here. One monomer comes and binds to the one half site and the other monomer comes and binds to the other half site, and that is how the receptor binds as a dimer to the target DNA sequence and activates transcription. And as I mentioned here, a glucocorticoid response element and estrogen response element differ from each other from two nucleotides. If it is AGA ACA half site, it is a glucocorticoid response element. If it is AGG TCA half site, it becomes an estrogen response element. And if you just make one amino acid mutation within the DNA binding domain of glucocorticoid receptor, instead of recognizing this sequence, now the glucocorticoid steroid receptor will now recognize this sequence. So, you can see how beautifully the nature has evolved target gene specificity of these steroid hormone receptors. Now, one can ask the question, you have glucocorticoid receptor, mineralocorticoid receptor, androgen receptor, they are all recognized in the same sequence, then how is the specificity brought about? Because the mineralocorticoid physiological effects are different, glucocorticoid receptor physiological effects are different, androgen receptor physiological effects are different, but they all seem to be binding the same sequence. So, how is it that the specificity is brought about? How specific physiological responses are brought about by these different receptors despite the fact that they are all binding to the same sequence? So, the answer is, although many receptors like GR, MR and AR, they all recognize the same sequence, the specificity is achieved by what kind of receptors are actually present. For example, a tissue which is responsive to glucocorticoids may only have glucocorticoid tissue which is responsive to glucocorticoids may have only a glucocorticoid receptor, it may not have a mineralocorticoid receptor. Therefore, it becomes responsive to glucocorticoid receptor. So, by expressing specific receptors in these target types, they can achieve specific target gene specificity can achieve. So, very rarely you will find that both PR and GR may be expressed in the same type of cells, or even if they are expressed in the same type of cells, the co-activators which are required for the activation may be present only for one receptor but not the other. We will discuss these things later in the lecture series. So, although the target gene specificity may be similar depending upon the cell type specific or tissue specific expression of receptors as well as different co-activated requirements, the different genes may be activated or different receptors may be activated in different cells. So, these kinds of experiments have been actually done again by very nice experiments where you can actually demonstrate these kind of specificities by actually, for example, in the case of estrogen receptor, how do you demonstrate estrogen receptor can actually activate only from estrogen response element but not from glucocorticoid response element in the same cell type. And you can actually show, you can take the estrogen receptor cDNA and transfer this in an expression vector and transfer this expression vector into the cells of your interest, and when the estrogen receptor is expressed, now if you add estrogen hormone to these cells, it will now activate only those genes which contain estrogen response element, but it will not activate transcription from those genes containing glucocorticoid response element, indicating that these receptors bind to very specific hormone response elements. That is how they bring out transcription activation. So, the identification of glucocorticoid receptors and deciphering the fact that these hormones go and bind to the receptor and then the receptor dimerize and they go and bind to specific hormone response elements, and the cloning of the glucocorticoid receptor cDNA as well as estrogen receptor cDNAs are one of the hallmarks or landmarks in the area of steroid hormone receptor signal transduction pathways. So, this marked a very important beginning in the understanding how these hormones function. This actually laid foundation for understanding the function of many other receptors which we will discuss in the next few classes. Now, what we discussed today is just about one class of receptors, what are called the type 1 receptors, namely the estrogen receptors, glucocorticoid receptor, mineralocorticoid receptor, progesterone receptor, androgen receptor. Now, there is now a huge family which is called as nuclear receptors family, and the cloning of these cDNAs and the demonstration that all these receptors actually have similar domain structure, and then people started using the zinc finger DNA binding domains of the receptors as probes, and using these probes they started screening cDNA libraries of various tissues, and they found that this kind of a domain structure is present in a number of other receptors. Okay? And this led to what is known as a nuclear receptor superfamily, and is a new nomenclature, and this nuclear receptor family includes not only the steroid hormone receptors, it also includes thyroid hormone receptor, retinoic acid receptor, what is called as the PPAR, and so on and so forth. And in the next few classes we will discuss some of these receptors and ask the question, what is their importance and how do they activate transcription. So, the the characterization or the cloning of the steroid hormone receptors is one of the very important landmarks in the area of nuclear receptor biology that actually paved way for the characterization of a number of other receptors which is mentioned here. So, the nuclear receptor superfamily consists of not only the steroid hormone receptors marked in red, but also a number of other receptors. And what is very interesting is that many of these receptors actually we know what kind of ligands bind, but there are many other receptors we do not know what kind of ligands they actually bind. For example, based on this, the nuclear receptors can actually be classified into again 3 different classes. One is the class 1 receptors, which we already discussed, namely steroid hormone receptors, which actually bind to palindromic sequences or inverted repeat sequences, and they are usually present in the cytoplasm in the absence of hormone, and they go into the nucleus when you add the hormone. Tomorrow, in the next class, we are going to discuss the class 2 receptors, which contains the receptors for vitamin D, thyroid hormone, and retinoic acid, and ask the question how understanding glucocorticoid receptor function have paved way for understanding the function of these receptors and what kind of sequences these receptors bind. There are also what is called as orphan receptors, that is, these receptors share the same homology with that of the other receptors in the content. They also contain zinc, they are also zinc finger transcription factors, but we do not know what are the ligands for these receptors, or some of them actually function as ligand independent transcription factors, and they also bind to different sequences. So, what I have tried to mention in this class is to demonstrate the discovery of steroid hormone receptors and how the discovery of steroid hormone receptor paved way for the discovery of other members of the nuclear receptor superfamily. And what I have shown in this slide is 3 important people who actually made a wonderful contribution for this steroid hormone receptor biology. One is Pierre Chambon in France, Ronald Evans in Salk Institute California, and Elwood Jensen, who actually made very pioneering work before the cloning of the receptors actually began. And recognizing their important contributions to this field, they were actually awarded what are called the Albert Lasker Basic Medical Research Award. This is often referred to as the American Nobel Prize. And it is many people working in this area believe that these people actually may deserve or actually deserve Nobel Prize, and they may be awarded Nobel Prize in very near in the future, because they made very important contribution for the discovery of the superfamily of nuclear hormone receptors and how these nuclear hormone receptors are important for a number of physiological processes. And I had the privilege of working in Ron Evans' lab, and so I was working in his lab for about almost 3 years, and what we worked is about what is called as a cross coupling of nuclear receptors with other kind of transcription factors, and we will discuss that later in this course. So, one of the people who made a very important contributions, I had the privilege of working with him. I just listed some of the key references which are listed here, which made a very important, which are very important, what are called the landmark papers, how receptors were cloned and how they were identified, and go through. If you go through some of these landmark papers, you will understand and appreciate some of the efforts that has gone into this discovery of steroid hormone signaling. I think I will stop here.